Okay, it's 1:05, so let me start. Hi, everyone. Thanks for joining my talk today. So this lecture is a part of our Fundamentals of Fluorescence Microscopy series. So today I'm going to give you an introduction to bioimage analysis. My name is Ellie Cho, and I am an application specialist for image analysis at the Biological Optical Microscopy platform at the University of Melbourne. So if you have any questions during this talk, feel free to type your questions in Q&A and I will answer them at the end of the talk. And also the recordings of this talk will be available on our website after this session, so you can check it out if you have missed any part of this talk. Also, I have put up a link on the chats, which I will also put up later at the end of the talk. And I'd like to start this talk with that the University of Melbourne acknowledges the traditional owners and custodians of the lands on which we work and learn, and their contributions of knowledge to our community. Also, I'd like to introduce Light Microscopy Australia, LMA, to non-LMA members. So LMA is a special interest group of Australian Microscopy and Microanalysis Society. And if you become a member, you get benefits like being eligible for travel bursaries, priority to data analysis workshops, and also you will get a regular updates of upcoming events, and you can participate the annual image competitions. So one of the big events, oh, sorry, I'm not really showing my slides up. Sorry. That was my slides. And I'll just turn off my videos. So yeah, back to the LMA. So one of the big events that's coming up next year is our biannual national symposiums in 2026, and it's in Sydney. And it'll be a 3-day symposiums and 2 days of workshops. And also this time we will be incorporating the OSBIAS, which is image analysis groups, and also IMOS, which is the intravital microscopy groups. And you can join the society at any time from this website. So in this talk, I will cover the essentials of image analysis workflow in 6 stages, starting from the planning, acquisitions, pre-processing, object detection, measurements, and reporting. I will also explore this with a couple of example workflows. So first, let's have a look at these 2 fluorescence microscopy images. The images on the left is the control group, and on the right is the one with the chemical treatments. And in measuring the effects of these chemicals, we can probably describe that the cells on the treated groups look slightly rounder in general, but this needs to be backed up by proper quantifications in science. So one of the useful quantifier for this case would be measuring the circularity of each cells. And the value of circularity would be close to 1 if it's close to perfect circle, and closer to 0 if the cell is elongated. Once we measure for each cells, we can then plot the values and run statistics. So that is pretty much the goal of bioimage analysis. So by performing analysis, we would like to obtain a quantifiable information from the microscopic image of biological sample. But this analysis always comes to the last step of our experiment, mainly because, of course, we will have to prepare the sample and take the image. And the thing is, with the microscopy and image analysis techniques becomes more and more sophisticated these days, we can get a lot of more informations. But the caveat is, at the same time, there are a lot more things that we have to consider. And the most common mistake is that we often neglect this complexity, ending up analyzing data incorrectly or repeating the entire experiments. So we need better planning. If you're planning to analyze your microscope images, the first step is to actually planning your quantifications, which means we need to determine what needs to be measured before starting anything. Then we need to determine the suitable microscope techniques that can generate images optimal for that particular measurement. Then we can prepare the sample based on this plan. So this planning should be done properly before any measurements begins. Well, sometimes we may not know what kind of feature we need to measure because the actual biology may be actually unknown. And this is exactly why we're doing science and research, right? So, and also even if we know what to measure based on the previous studies, the actual observation may require different measurements. So the idea is we always need to carry out a pilot study before undertaking a full-scale experiment. And then from the pilot experiments, we will be able to optimize settings, readjust our plans iteratively, and also determine the final sample size. And once optimized, you can then run full-scale experiments. And if you're not sure what's possible or don't know where to start, it is also a good idea to consult with your local core facility staffs, or you can contact us at your planning stage. So now let me get to the second part, introducing the general workflow of bioimage analysis. So let's say we have acquired a raw microscopy image like this. Generally, image analysis workflow involves first pre-processing of raw microscopy image to correct any defects happens during acquisitions, and then segmenting objects or defining a region of interest before measuring any quantifier and reporting the result. And depending on the type of analysis, we can either use a single software package that covers the entire workflow or use multiple software to access the specific functions. And there are actually many image processing software tools available. So finding the best tool can be quite challenging. And there is a search engine available like here, which was developed by the Network of European Bioimage Analysts, Nubius. And in this website, we can get curated information of image analysis softwares and which types they are. But if you look at the number of software curated here, there are currently more than 1,400 softwares. So this database is helpful if we are looking for some specific analysis tools, but it may be too extensive, especially if you are new to this area. There are also publications reviewing these softwares. So for example, one of these papers compares the functionality of open source softwares. But when it comes down to analyzing images for your own research, and if you haven't done any image analysis before, I'd recommend you start with Fiji because Fiji is the built-on ImageJ and it's most widely used, community-driven, open source software, and also give you access to hundreds of plugins. So now we can get to the first step of image analysis workflow, which is acquiring microscopy image. acquiring proper image is really crucial in getting the proper measurements. So because the basics of microscopy was covered in our previous lectures, today I will cover the 5 key factors that affects the most on image acquisitions. So these include resolutions, sampling rate, bit depth, dynamic range, and saturations. I will go through one by one on the following slides. But to understand the basic requirements of acquisitions, first we need to know how the microscopy image is generated. So let's say that these are the fluorescence signal of our objects. And when we use the microscopes, this signal will be formed as an image through the optics. And this is an analog and continuous signals. And when the image is detected on our camera and saved on the computer, the signal has to be converted as a digital format. So pretty much the image we get is the digital image that is composed of many pixels. And the pixel is a unit of our image, and each pixel has a defined size. So the first step of control these image resolutions is, of course, using the correct objective lens during the acquisitions. And if we use the wrong lens, for example, in here, the analog image won't have enough resolution to start with. Of course, then its subsequent digital products. And how the lens determines the resolution was covered in our first lecture, so you can revisit this if you want to learn more details. But the second step that controls the resolution is using the correct sampling rates when we convert our analog signal to a digital format. So what we want is, if we want to use the correct sampling rates, then we need to follow a Nyquist rate sampling rate. So the Nyquist sampling theorem means that to get the proper representation of our data, you need to sample it at least twice the highest frequency that's present in the signal. So what does that mean? Let's say that these black curves is our signal, and the red dot here is where we will be sampling. So if we say sample 1 point per cycle, then the sampling results will be like this, which is not really a proper representation of our data. And if we increase the sampling rates, that improves it. But when we sample it at least 2 times of its signal frequency, then it starts to reflect the shape properly. So this sampling rate of 2 is pretty much the best minimal requirements we need to use. In microscope image, the Nyquist conditions translated that the pixel size should be at least 2.3 times smaller than the object you are trying to resolve. Which means, for example, if we try to resolve a cell with a diameter of, say, 10 microns, then your pixel size should be at least 2 times smaller than that. And the factor 2.3 accounts for many different factors, such as optical diffractions, and also the shape of pixel is being square. So this is an example where the objects were taken with Nyquist sampling rates. And if we use lower sampling rates, which means larger pixels, and this will result pixelated image like this, and we will lose the details of our shapes. On the other hand, if we use higher sampling rates, this will generate prettier image, but this can be also impractical as the imaging will take longer and the files will be larger, which means the data processing will be slower. So in summary, what we want is we want sufficient pixel resolutions while avoiding under or oversampling. So next consideration is a bit depth. As I mentioned, microscopy image is a bunch of pixels, and these pixels have its own intensity value as well. So for example, these pixels on this image has intensity value of 159. So what does that actual value means? So when we convert this analog signal to our digital image, we can choose on how to convert the strength of signal into an intensity value. So this can be converted simply as 0 and 1 if we are using a 1-bit depth image. Or it can be a 4 gradient steps if we use 2-bit depth. Or 8 steps in 3-bit depth. Or 16 steps in 4-bit depth. And we can increase that way, and at 8-bit depth, the signal can be addressed in 256 gray values. And this gives smooth enough gradients for human eye. And that's why many microscopy images used to be saved in this 8-bit format. And this is good enough if the image is used only for visualizations. But for quantification, we are much better off to use a higher bit depth, such as 12 bits or 16 bits, because it records the intensity with over 4,000 and 65,000 grayscale. So if you look at this example, that image was taken on 8-bit and 16-bit. For visually, we won't be able to see the difference. But if you actually start to reading the pixel intensity, you can see that the higher bit depth can record the difference more in details. So then we will get more accurate quantifications. So for quantification, we need to use higher bit depth, more than 8 bits. And the last two considerations are the dynamic range and saturations. So the intensity distribution of this digital image is often presented as a frequency histogram like this. And this histogram is very useful for overviewing the intensity distribution of the image. So here, this is the histogram of this green image. And you can read the histogram as there are a large portion of background here, while the mid-intensity and bright parts are relatively well spread out. In the example on the left, you can see that the total range of the histogram is skewed to the left because the image is generally darker. And in this case, the signal here is not well separated from the background. So this image is less informative, and this can result poor quantifications. So during our acquisitions, we can check this histogram and adjust the image settings to use the whole dynamic range as much as possible. On the other hand, too bright images can result saturation. So the saturation means that some signal has reached or exceeded the maximum detection range. And when this happens, we won't be able to get the actual intensity in that region. So the quantification is no longer accurate. So this should be avoided during our acquisitions. And most of acquisition system provides a range indicator as well as histogram. So you can sort of see the saturated pixel visually and avoid it. And there are a few other things to remember during acquisitions is that the images we are going to compare should be acquired and processed using the same setting so that they are comparable. And also when saving the images, we always have to save as a raw microscopy format or at least the OME-TIFF to preserve the metadata. So metadata is really important for quantifications. And also we should avoid saving as a standard image format such as TIFF, JPEG, and PNG. You can save or export your data from the raw microscopy formats, but once it's converted, you will lose the metadata, which can be quite crucial in your quantifications. Okay, so let's have a quick break and test if we remember some important factors of acquisitions. So the first question was, what is the ideal pixel size for microscopy image? So if you remember the Nyquist rate for the images, we want that to be 2.3 times smaller than my objects. So it's good that most of people have got the correct answers. And the second question was, which bit depth should you use for intensity measurements? Yes, the 16-bit or any bit depth higher than 8 bits would be preferable. And most of you have got the result, correct answer, so that's great. Right, I'll stop sharing. So within these 5 next sessions, I'll have the pop quiz after each section. So hopefully we can sort of reiterate the contents. Okay, let's move on. By the way, this cat picture has nothing to do with the questions, so. Right, so the next step of workflow is pre-processing. So this one's, even if we have managed to take the best possible image by following the rules that I mentioned before, the quality of raw microscope images can still be compromised. So in many cases, we need to correct it afterwards. And there are many, many different procedures out there. So covering everything will be impossible. But in this talk, I will introduce 5 most common correction procedures. And the first thing to consider is denoising. So every microscope image contains a noise that are mainly coming from the electronics of the acquisition system. So this noise can be a problem in object detections. And the common options to reduce noise is using image filters, such as median filter or Gaussian filter. The median filter replaces each pixel intensity with the median value of its surrounding pixel. So if you were to apply a median filter from the image on the left to the right, then you can see the middle pixel intensity is now changed and grabbing the median value of its surroundings. By doing this way, what it does is it preserves the edges better without changing the value too much. Whereas the Gaussian filter applies this Gaussian convolution. So it's adjusting the intensity value that fits into the Gaussian shape. And as a result, it smooths out the signal. And also this is used to blur out the signal if it's necessary. So it's apparently doing slightly different things. And also recently there have been many deep learning approaches that was developed for denoising the microscope images. And the two good examples are called CARE, and another one is noise to void. So the CARE method, it requires the noise image and a clean image as a pair, so that it's try to mimic the clean image by adding by submitting your noise image. Whereas the noise to void is trying to learn the noise pattern from the noise image itself and try to remove from there. So it's unsupervised. Another pre-processing tools is the background corrections. So as you can see in this image, the center of the microscope image often appears a lot brighter because of optics or misalignment, and this can affect the object detections a lot. So of course, the aligning your microscope will help, but there may be still some residual unevenness even after that. So then we can apply some algorithmic corrections. So one of the common correction tool is called rolling ball background subtractions. And this estimates the background as a large ball shape and based on the user-defined value, and then it can subtract from the original image. But sometimes we may have even more complex unevenness. Then we can use a flat field image. And this flat field image is pretty much an empty background image that is captured with the same microscope and same settings. And then we can use this to correct the original image by doing a divisions. And sometimes there's some stripe artifacts, and this can be corrected using a bandpass filter. And this bandpass filter removes the unwanted frequency components while it's still preserving these image details. There are also many other more advanced background correction tools. One of them is called Basic. It's the name of the plugin. And this computes the shading metrics and corrects the shading. And this type of background correction tools are available in most of image analysis programs, such as Fiji or Cell Profiler. The next one is bleaching corrections. So in fluorescence imaging, this type of intensity fluctuations can happen because the actual fluorophore bleaches over time, or fluorescence lamp itself can jitters. And this is an example where the image was taken over time and it's shown as a montage like this. And you can see that the intensity of the sample bleaches, and this bleaching makes the image analysis very difficult. So in this image, those decreased intensity by bleaching was fitted by exponential decay equations. And we can correct this using Fiji plugin. Sometimes not everything may fit as exponentially, and the bleaching may happen, which we can also correct different algorithm called histogram matching. And there are also few tools available to correct such bleaching. So it's called the bleaching corrections or bleaching corrector in Huygens. And also things that we have to be consider is that this type of correction is really useful for segmentations or detecting the objects, but it should not be used for intensity quantification because pretty much we are making lots of changes in our intensity. And the another artifacts is the image misalignments. So this misalignment can happen in multidimensional data. This can be in X, Y, or Z, or over time, because mainly from the drifting of stage or sample movement, as you can see in this example. So this drift can be corrected for different type of plugins before any detection or measurements. Also, this type of misalignment also happens between different fluorescence channels. And this is because of the chromatic aberrations. What it means is the different color, different wavelengths focuses at slightly different positions. And this effect becomes more noticeable for very small objects, even with the calibrated lenses. And correcting this aberration is especially important for colocalization analysis. And tools like a Transform J or chromatic aberration corrector can help with these chromatic corrections. Another case when we need alignment is that when the images were captured at multiple positions to form a large mosaic image. Then these individual images should be stitched into a complete image before analysis. And Big Stitcher, for example, is a powerful plugin in Fiji that also can handle large files. And the last example of this correction method is called deconvolutions. So deconvolution is pretty much a mathematical image restoration method where we can estimate how this light scatters on our microscope settings. And once we estimate, we can restore the signal back to the original as much as possible based on these calculations. So by doing these calculations, we can increase the image resolution in all directions X, Y, and Z. And as you can see in this example image, if we were to measure the localizations of this green and red channel, the measurements will be much more accurate after deconvolution here. So the deconvolutions during this process, it also increased the contrast and also remove lots of noise, as you can see in this example. So altogether, these deconvolutions can improve the quality of data visualizations as well as analysis. And the most commonly used commercial softwares is Huygens, and it is available at our platform. So if you'd like to access it, feel free to contact us. Okay, here's another break. Let's check our understanding on our pre-processing. Was which filters can be improved during pre-processing step? So the correct answer is we can increase the signal to noise ratio as probably mainly using for deconvolutions. And yes, we can, of course, correct the uneven background illuminations. I've shown you 4 different examples. And another right one is the resolutions. That's again through the deconvolution step. The things that we cannot really change during the pre-processing is the desaturations. So this is kind of related to the previous topics on the acquisition parameters. Is once the pixel has been saturated, we cannot get any further information from there. So this is strictly has to be avoided during your acquisition step. And also we cannot improve the bit depth. Same way, if we have taken all these signals into 8 bits and the grayscale is only 256, we cannot make it into 4000s, which will be just simply empty amplifications. So yeah, saturations and bit depth has to be controlled during the acquisitions. You can still see most of people have got the right answer, but this question can be a bit confusing, so understandable. Okay, let me just stop sharing. And we can move on to the next step, which is the object detections. So when detecting an object from our image, the simplest way to find an object is, in this case, is finding it as a point using local maxima. So it's looking at the intensity and find where the local maxima point is and finding it as a point. And this is really useful for quick counting, but it doesn't really give us other information such as size or shape. So the object detection is usually done in multiple steps. The first step is classifying the pixel as signal and background, and then converting them into a binary image, black and white. And this process is called segmentation. And this segmentation is really crucial and also challenging at the same time. So there has been many different approaches developed, which I will cover in the next slides. And once we segment, then we often need to do some additional title process. So in this case, splitting touching objects. And then we can finally group the pixels into individual particles. So let's have a look at the segmentation process. Out of 3 that I will cover, this is the conventional process. So in conventional segmentation process, usually what we do is we set an intensity threshold to separate the signal from the backgrounds based on the histogram. And as a result, this will generate a binary image like this. But the problem is when using a different threshold value, it can also give us the completely different result. And this is problematic for measuring area. So the simple approach is using a negative control image where no expected signal. But in this method, the consistency is the key. So we need to apply the same threshold to across all the images if we want to be objective. But in many cases, the background level can vary different. And this makes the single thresholding method very ineffective. And so this requires adaptive method for better accuracy. So to compensate such issue, we can use auto threshold method, where the threshold value is calculated logically in each image. There are many different auto threshold algorithms available. And for example, one of the method called isodata finds a threshold value where the average value of the pixels above and below the threshold are identical. So this is where it finds the threshold based on this histogram. Another method called intermode, it assumes the histogram to be always a bimodal, and it finds the 2 local maxima, M1 and M2, and computes the midpoint of those 2 points. So because this one method may work better than the others depending on the type of images and type of your histogram, what we need to do is test which method will work best for our image. So for example, in image J, we can test 17 different auto-threshold methods and also 9 different auto-local threshold methods in one step. So then we can browse the result and find the best method for our image. And once we have found this, the idea is to use the same auto-threshold algorithm across all the images. So in this case, we're kind of assuming that the entire data set will have a similar type of histogram, while the actual range may vary a little bit. And this type of conventional threshold approach works well for fluorescence images, but it also struggles with low contrast signal or non-fluorescence images like bright field, color image, or electron microscopy images. So, and in this case, machine learning can help. And in this machine learning approach, what we do is usually users annotate the pixels and they can assign pixels to different groups manually, usually using this type of brush tools within the program. And this manual annotation is fairly simple and interactive. And then what it does is the software analyzes the many different features of these pixels. So it's not just looking at the intensity, but it looks at the color, texture, and also the surrounding context by applying many different filters. And then it calculates the category likelihood for each pixels. Then it will generate a probability maps and sometimes a labeled image like this. So there are many software providing this type of techniques like Fiji, Elastic, CubePath, if you're looking for freeways, but also commercial programs like Imaris, Zen, Avia. And most of this software interface, as I mentioned, is user-friendly. And the good news is no programming skills is required. So this is fairly accessible techniques these days. But in image segmentation, there are still many more complex patterns of it, that's challenging us. So there has been many deep learning models developed. So for example, the method called UNet can segment individual cells from this tricky image, such as bright field, like this, into the individual cells. And another method called STATIST can detect these round objects, such as nucleus in biological sample, really well. And it also works on both fluorescence and histology images. Another really popular deep learning method is called CellPose, and it's a really powerful tool that can detect various shapes of cells. So the thing is, most of this approach requires large amount of manually annotated data and also programming skills for training the model. But these days, there are a lot of pre-trained models available, which we can access without particularly learning the programming skills. So for example, the Bioimage Model Zoo websites here, they provide a curated deep learning models. So we can just go and browse through and find a suitable model for our image. And then what you can do is you can download the models and then use it through the user-friendly softwares like Fiji, Elastic, CubePath, and Cell Profiler. So after those either conventional machine learning or deep learning steps, once we have segmented the pixels using those and have the binary image, as I mentioned, we can refine them with more morphological operations. So for example, we can separate connected particles using watershed algorithm. And we can also create a distance map like this, which means that it assigns the intensity incrementally from the object borders. But also we can use some Boolean logics to filter the objects from the other images. So in this case, it's using the Boolean logic AND functions and try to find the spot that's only inside these objects. Then you can use that through the binary image processing. So I will cover some of this processing through the example workflows in the next part. But because I've covered all the object segmentation parts, let's also check our understanding of these sections. So the question was, in conventional segmentation, what is the best way to set threshold on data sets with various background level? So most of people have selected the correct answer is applying the same auto threshold algorithm. Yeah. So we don't want to apply the same threshold value, which is a single value that may not really work on the images with different background level, and apply different threshold value manually. It's it just include lots of subjectivity, so it will be really hard to explain how you actually end up choosing the values. So it's best to be calculated logically. And the third option was apply different auto threshold algorithm for each image. Again, this is not really considered as the objective options. So the best way is to find the algorithm that works your entire data set and use that auto threshold method. Okay. So now finally we are at the second last stage of bioimage analysis, which is measurements. So these are the most common measures, and I will explain them using some example workflow in the next slides. So the first one is number, which is pretty much counting objects. So one of the common workflow is counting positive objects. So in this example, we have two channels where the first channel shows the every cells on the nucleus staining. And the second channel shows the phosphorylated histone staining, which is the marker for detecting mitotic cells. And if we want to count the percentage of these mitotic cells, then of course we first have to detect individual objects for each staining. And in doing that, what we can do is we can also remove the objects that are touching the borders, because often they are not complete informations. Then we can filter the nucleus objects based on its presence of mitotic cell marker. So if it overlaps with this marker, then we count as 1. If it doesn't, we count as 0. So this is fairly simple workflow. And what we'll do is then we can pretty much calculate the total populations and positive populations, and we can plot in a graph. And also, it's also a good idea to show a proof image like this so that we can compare if our measurement has been correct or not. The next common measurement is the intensity. So if we like to compare, say, the brightness of the objects in these images, then the mean average intensity would be the right choice, because the mean intensity is the sum of total intensity value divided by the area of the object. So this represents the standardized intensity regardless the size of the object. And that's why the mean intensity can be used as a general measure of staining. In this example, so for example, if we like to measure how much of protein has translocated to the nucleus from the cytoplasm for these images, and the one staining is a nucleus staining to define the nucleus area, and another is for the protein of interest staining to measure the actual intensity. And using both images, we can define the nucleus and cytoplasm area of each cells, and then measure the mean intensity of proteins on the nucleus and the cytoplasm and calculate the ratio as an indication of translocations. So the mean intensity is the most widely used intensity measurement, but apart from that, we can also measure other intensity features like minimum intensity, maximum, median, sum, or standard deviations. And although they are not used often, they can be useful for measuring specific features. So as an example, these two objects in the image have similar mean intensity, but we can clearly see that there are differences like left cell staining is smooth, where the right cell shows more intensity fluctuations. And also the structures are more granular. And in this case, if you look at the histogram of each image, the intensity distribution is more widely spread on the right cells, and we can measure these dispersions. So the standardized measure of this is coefficient of variations, which is the ratio of the standard deviations to the mean. So this value can be the measurements for the degree of fragmentation or homogeneousness of the intensity. And the next common measurement is the shape. So biological structures can be measured in many different parameters. And let's have a look at these images, two images, that we can say that their shapes are quite different, but which measurement will be the best to represent the differences? At first, we can measure the size using area. But the actual measurements, they are not very different. And in this case, we can see the right cell is more spread. So then what we can measure is the ferrite diameter, which is the longest possible line that can be drawn on the objects. Because they are more spread, this will give a higher value. But also for these cells, the total surface area is a very important biological measures. So we can measure the parameter, which is an outline of the object. But when you look at it, they also have a very similar parameter measurement. And in this case, different type of measurements. One of them is a convexity, can be a good measurement as it's measuring the ratio of object parameter to its convex hull parameter. So the convex hull is the minimal polyhedron fits. So it's like a shape when you put a rubber band around the object. So you can see the convex hull for these cells in the yellow line here. So if we measure the ratio of its own parameter to its convex hull parameter, then we can measure more complex shape parameters. So in this case, the right cell has lower convexity value, and which indicates the more irregular and rough in shape. So there are many different shape measurements available, which we can't, of course, cover everything today. And you can find more details in these websites to start with. And the next common measurement is distance, especially the distance between objects. One way of doing it is measuring the nearest neighbor distance between the centroid of an object. And usually this method requires less computations, and it works well when the features are small compared to the distance between them, and also when they are relatively round objects. But if the objects are large compared to its distance, and also particularly when they're irregular in size and shapes and have a very different orientations, it may be more appropriate to measure the distance from edge to edge rather than between the centroid. So Here's an example where we want to measure the relationship between the objects. And in this example diagram, the left image represents a cell that are receiving a synaptic input. And on the right image, we have a synaptic protein staining for the whole tissue. And if we want to know how much synapse there are for these cells, we will need to detect the particles that are touching the cells. So how do we measure this? We can do this by first generating a distance map outside the cells. As I mentioned briefly, this map is an image where its pixel intensity increases gradually from the border of the cells to the outside. Then we can measure the intensity of this distance map for each particles. And the particle with the minimum intensity of 0 means its distance from the cell border is 0. And higher value means it's away from the cells, etc. So based on this, we can filter the particles based on its intensity to the distance map to detect the cell, the one only touching the objects. And also when it comes to the close relationship between measurements, colocalization analysis is one of the most popular methods. I'd like to start with that when we are referring the term colocalizations, we should be very careful not to interpret this as two molecules are on the same locations. So in bioimage analysis, colocalization analysis generally gives an indication of the two fluorophores are generally near each other. And this doesn't indicate that the molecules are interacting or in the same locations. So when we measure the colocalizations, we can first measure if there's any relationship between the channel intensities by looking at its correlations, as in whether the brighter green correlate with brighter magenta or red, etc. And this is, we can measure using the Pearson's coefficient. Or other common measurements is measuring the co-occurrence, which means whether the fluorophores are generally near each other. So in measuring this type of co-occurrence, we can measure Mandel's coefficients, which is measuring the sum intensity of the overlap proportion to for each channel. Or we can also measure the overlapping portions based on the object. And also when it comes to the colocalizations, it is really important to consider that this type of quantification is heavily dependent on the resolutions of the microscopy images. So as you can see in this example, that the one on the left is taken with the lower resolutions, and on the right is the same type of cells taken in higher resolutions. So you can see that the low resolutions will end up giving you unnecessarily higher colocalization measurements, which can be misrepresenting the true nature. So we have to be very careful in planning this type of measurements. And also the last common measurement is tracking. So when we have a time-lapse image like this, we can measure parameters that are specific to tracking, such as speed, how fast they are, displacements, how much they have moved, accumulated distance, how much in total they have moved, and directions, orientation, straightness, and the duration, etc. And for example, if we want to investigate the mobility of the cells, the displacement chart can be a good visualization tools as well. So in here, you can see that the cells on the right, the movement has been reduced after drug treatment. Okay, so that's the end of the measurement part. So let's again check our understanding on the measurements. So the first question was, what is the best quantifier for comparing the brightness of microscopic image? Yeah, most of people have got the right answer as a mean intensity. And that was an easy one. And the second one is to select the correct description of colocalization analysis. So this one, there's bit of differences. So what it does is the first answer, it measures the interaction of two molecules. Now, as mentioned, it's not really measurement of the molecular interactions. So if someone wants to measure the interactions, will have to be we'll have to use completely different type of microscopic experiment like a FRETs, where it actually signals turn on and off based on the molecular interactions. Because when we just describe as a standard pixel-based colocalizations or object-based colocalizations, we are really limited by the size of the pixels and the resolutions. So it's more like indirect measurement of that fluorophores are close together rather than the actual interaction, not the functional one. Yeah. And yes, it measures the correlations of the pixel intensity. Yes, that's correct. If we use the Pearson's correlation coefficient, and it measures the general proximity of molecules, exactly what I just described now. And yes, colocalization measurements on low resolution image may misrepresent the data sets. Yes, I think most of people have got the correct answer. So thank you very much for that. Let me stop sharing. And then now we are in the real final steps of bioimage analysis, which is reporting and documenting. So since, as you can see, the most of workflow involves multiple steps, so it's crucial to record these processes and parameters, not just for your own reference, but also it helps others understand and also to be able to reproduce your work. So this is one of the published example where you can see the analysis process is clearly documented as a flowchart. So based on this, we should be able to follow pretty well. So some programs like Cell Profiler makes it easy to build this type of workflow directly within the program. So here we can add and remove individual analysis modules, and we can create our own custom pipelines. And once we're done, then this pipeline can be used for batch analysis, but also can be saved as a file, which means it's easy to share and reuse, which is great for reproducibility and collaborations. And if you go to their websites, they also provide many good example templates, which are really helpful for learning how different workflows are structured. Some pipeline environments, so this type of environments is not available in other popular programs like Fiji or QPath, but on these programs, the workflow can be recorded via script recorders and applied for batch analysis. And there's another software where you can automate analysis by scripting, include IC, Python, MATLAB, KNIME, and more. And this script is also a very important recordings of your publications. And the thing is, it's really good to have this many options, but at the same time, I'm saying not every biologist needs to be a programmer. So if you like to automate your analysis, or if you want to get further help on image analysis in general, you can always contact your local image analyst. Here's a link for the Australian image analyst websites, or you can also collaborate with our platform. So when we report our measurements, what we do is we often skip over the details of image analysis workflow, which kind of make it hard for others to reproduce a result. So many of you also have come across the papers with interesting method, but couldn't replicate those because there's no details. And sometimes the only description is something like image was analyzed in Fiji. So there's a lot of problems for that. So to address these issues, the bioimage analysis community have developed a checklist, and that was published in Nature Method 2 years ago to improve the reproducibility. So pretty much if you are publishing a new workflow, what you need to do is you should clearly describe each component and the sequence of the steps. And also you should upload your workflows and sample data to an online platform so that people can try out. It is also highly recommended to explain the rationales and the limitation of the workflow. So not just showing the good things, but what it cannot do could be also very useful information. And ideally, they're asking to include any screen recordings or tutorials. So this helps others to adopt to your method. And of course, you can then increase the impact of your work. So this checklist also covered the best practice for publishing microscopy image. So if you can, please read it through. So which will be really important. Okay, so the last part was really short, and let's check our understanding on our reporting for using the final all options. So here we've got some interesting things happen. So the question was, what needs to be included in reporting new analysis workflow? And originally, I set the correct answer just 2, which is the detail of individual components used, and also example data and code. But a lot of people also answered of dates when the workflow was created. And I guess it is actually correct in some senses, especially when you think about the version control. So yeah, I might just change the answer a little bit, but yeah, I guess most of the time, the more information, the better. So I'll still take it as a yes. So let me just stop sharing. So well done. Okay, so I'm just wrapping up this talk. So here are some great resources you can access if you want to learn more about bioimage analysis. So the first is our platform websites, where we host materials from our previous workshops, including this lecture series and also the Fiji workshops. And it's also regularly updated. So please check here often if you like. And the second one is Image SC Forum, which is pretty much the first place that I would go to whenever I want to search for tools, workflow, recommendations, and troubleshooting. It's also a very active forum, and it contains really useful information. So here I put as a long address, but just type it in image.sc on your browser, then you should be able to access it. And also finally, each software tool usually has its own websites with manuals, tutorials, and example workflows. So these are also very great resources for learning specifics. So this is the end of my talk today. So thank you very much for your attention.